Hello, everyone. Welcome to today's webinar. We're going to start in a minute. We're just letting people into the room. And we're here today with the speakers that we're going to introduce themselves in a second. And today's host is going to be Sandra Pavlovic from Rare Connect and Melanie Temple also from Rare Connect. Sandra?
Okay, I guess we can start with. First of all, good day to all. I'd like to welcome all the participants that has joined this Bingo and Rare Connect joint webinar today from different countries and time zones. My name is Sandra Pavlovic and I'm the Rare Connect manager. And today with my colleague Melanie Temple for this webinar, we are the meeting hosts. Just a few words on housekeeping. As we all have used to these new conditions and online environment since the pandemic started, I won't take time to present the tool we use today in details. However, in terms of a more technical aspect of the meeting, instead of organizing a classic webinar, we are holding an online format meeting. We actually thought it would be more convenient to participants if giving you the option to unmute your microphone and speak up during the time set for discussion. As for the classic webinar setting, this option isn't available though. Please keep in mind that this webinar is recording, so you can turn on the camera, or if you don't feel comfortable with, you may stop the camera, of course. It's not certainly mandatory, however you feel more comfortable with. You are able to ask any questions or write comments you may have via the chat feature along the way. Please just make sure you have selected in the chat box everyone, so we don't miss to address any of your questions afterwards. This is the meeting agenda, and before I introduce the speakers, I'll say a few words about Rare Connect, since some of the participants here may not yet became Rare Connect members, therefore not familiar with our platform. We will start with the first presentation presented by Dr. Kate Baker with the subject research after a genetic diagnosis. What are the questions? And right after, we will hear presentation from Dr. Anna Kolesnik-Taylor with the subject studying rare genetic disorders, what is known about the brain and behavior, and what is the future of research. When we hear from our speakers, we will invite Mr. Paolo Mancerini to join us and share his experience as a father who participated in the Bingo study. After we hear all the speakers, we will start with question and answer session, where you will be able to ask any of your questions either through chat box, or actually this would be a perfect time to unmute your microphone if you wish to ask your questions or comment vocally. If you think of any questions during the single presentation, please write them in the chat, and we will bring them after each presentation finishes or after all speaker finishes their talks within the time dedicated for the Q&A. As many of you already know, Rare Connect is a multilingual platform consisting of more than 260 rare disease-specific communities, enabling thousands of people affected by the rare conditions and those who care for them to connect to each other, no matter where they live or which language they speak. Patients and their families use the Rare Connect platform to share their experiences, exchange disease management techniques, better identify symptoms, or just empathize with one another. Given the low proportions of sufferers in each country, connections across language barriers are especially important to patients. Rare Connect notes that information about rare disease is primarily published in English, so non-English speakers have fewer opportunities to learn about and ultimately find ways to manage their conditions. Breaking the language barrier has thus been a priority for Rare Connect since its founding, and today Rare Connect allows patients from different countries to interact in 13 languages. These communities serves as a kind of a support tool for all those patients who might feel isolated or don't have the support they need in their countries. For the families, this webinar is intended for. Rare Connect have CIT1 associated neurodevelopmental disorder community, also DNM epileptic encephalopathy community, and trial-related intellectual disability community, for which conditions this Bingo research project has main focus on to find more about your research priorities. So just a short introduction of the speakers' bios who we will be hearing in a minute to present us a Bingo research study, its values, purpose, and opportunities. Dr. Kate Baker is a program leader track at the MRC Cognition and Brain Sciences Unit, University of Cambridge, where she leads the Genomic Disorders and Cognitive Development program. She's also a honorary consultant in clinical genetics at Cambridge University Hospital and an affiliate PI of the Academic Department of Medical Genetics. Prior to this, she completed her medical and research training at Oxford, UCL, and Cambridge. Our other speaker is Dr. Anna Kolesnik-Taylor, and she is a research associate at the MRC Cognition and Brain Sciences Unit 2, and she is a new addition to Kate's team. She completed her undergraduate in psychology at University of Sheffield in 2015, and her master's and PhD in developmental cognitive neuroscience at Birkbeck College, University of London, at the start of 2020. Thank you for joining this webinar, and hope all the parties will find it very helpful. I'll now stop sharing the screen and pass the floor to Dr. Kate Baker to introduce the Bingo research project and the research team.
Thank you so much, Sandra, for that lovely introduction. And thank you everybody for taking time out to join us today. It's a real pleasure to see so many people. And also a big thank you to the Rare Connect team for all of your work with us over the last few years. So I worked particularly with Marta, who was here before, to set up the SYT1 connected community. I know how much that has meant to the members of that community and to me as a clinician, as a researcher as well. So thank you. Can you see my screen now? Okay, that's wonderful. So I'm just going to spend about 10 minutes, I suppose, giving you a little bit of the context to the Bingo project. And do feel free to stop me, interrupt me if you'd like to, and there'll be some points for interaction as well. I can't see the chat right now, so maybe Anna, if there's a question in chat, then you can maybe unmute and let me know. So I don't need to introduce us. Sandra has already done that. Thank you so much. So I'm going to go straight to really the point of why I established this research project quite a few years ago now. Why did it begin? Well, I'm a clinical geneticist. And those of you who are here as patients or as family members and carers or as professionals may have some experience of clinical genetics. So this is how it's supposed to work in the modern era of genomic medicine. So a patient walks into the clinical genetics office or department. They bring all of their medical history in a rucksack with them and their genome, and me, the clinical geneticist, can rapidly make a clear genetic diagnosis for that person and that family. And it shouldn't take very long these days. Of course, it still takes a long time. And then with that genetic diagnosis, the door to a bright future is supposed to open, where this genetic diagnosis provides a really thorough and complete explanation of the problems that this person has had in the past and is happening now, and that this genetic diagnosis will really match up very well with the needs of that person. And that there'll be very clear information about the chances of the same problems affecting other members of the family, that's recurrence risk, and that this genetic diagnosis will give very clear prognostic information. And we mean by that very clear guidance about what kinds of medical problems this person may experience in future, how their cognitive development will proceed, whether they will have future mental health difficulties, and really what the landscape of their future journey is going to be with this diagnosis. And as well as that information about future prognosis, the hope is that a genetic diagnosis will lead to changes in treatment and support for that person, which alter their life course and their outcomes. And that treatment might be directed at the gene itself, or at the effects of that gene on biology, or in different ways of supporting that person through education or through occupation in the rest of their lives. So that's how it's supposed to be. So I wonder, I don't know if you can use the reaction buttons for those of you who've experienced clinical genetics, or where you have a member of your family who's received a genetic diagnosis. I guess put your thumbs up if your experience of genetic diagnosis has matched this promise. Or put your thumbs up if it's fulfilled some of this promise, but in fact, where a genetic diagnosis has generated a lot of further questions and perhaps some frustrations alongside that. You can wave your hand or use your thumb box if perhaps it hasn't been quite like that. So I would say, as a clinical geneticist making genetic diagnoses, I do hold on to the hope that genetic diagnosis can answer all these questions, or some of these questions some of the time, but at the moment we can't. And that is a very frustrating position to be in. So it's great that we can make genetic diagnoses, but we lack so much of the knowledge and information that needs to come after a genetic diagnosis to really help to make genetics make a difference beyond just finding a cause, moving beyond just finding a cause, which is very important, to making that cause really useful for patients and families. So it's that hope and also frustration which motivates me in my research and is the heart of the Bingo project. So I've asked, what was that like for you? And I'm perhaps at the end of this presentation, or perhaps if we stay in touch, you can tell me more about what this experience has been like for you and your families. So what do we want to understand in very simple terms? So what we do is we want to collect information about groups of individuals who share the same or similar genetic diagnoses related to neurodevelopment, and map the similarities and differences between them. So very basic information, what is it that's the same between two people who share a trio variant or an SYT1 variant, but also importantly, what isn't the same, what's different, in what ways do people vary within the same genetic diagnosis group? And that tension is really important, because I think that understanding both similarities and variation gives us a really clear handle on what aspects of brain development and cognition are really affected by these genes. And also that understanding that variation gives us really clear, some potentially clear insights into how to better support people in the future. So that's what we want to do, and why we want to collect such detailed information. And we've done that over a number of different single gene groups, but also we group together different genetic disorders, and Anna will talk to you more about that. What else do we want to understand beyond just mapping variation within genetic disorder groups? Well, genes don't affect cognitive development, learning, emotion regulation, social interaction, just by direct relationships between genes and behavior. It's got to have something to do with the brain and brain development. And there's such an amazing opportunity to understand more about fundamental aspects of brain development by working with people with genetic diagnoses, and also that by understanding aspects of brain development, we'd be in a much better position to get under the surface and help each person by understanding what actually drives their learning difficulties and their difficulties in everyday life, going beyond the surface mapping of behavior to actually understanding what the specific problems are at the level of brain function. And we do that by sometimes looking at MRI scans that patients might have had at their base clinical centre, or sometimes people come and visit us in Cambridge for scans, MRI scans, or clever scans called MEG scans. But increasingly, we have some almost like intermediate technology tools, so some ability to come and visit people and do EEG data collection at home. This is actually my daughter here and my son taking part in some pilot studies, where we can try and get a better handle on what aspects of brain electrical activity are typical or atypical in individuals with genetic disorders when they're in their normal home environment. And then the third thing that we want to understand very broadly is going beyond the individual person who has this genetic diagnosis in a family to understand actually families as a whole. So there's most of the focus on genetic disorders research tends to be on the diagnosed person or the patient, but they don't live in isolation. They live alongside brothers and sisters, mums and dads, step-parents, grandparents, friends, neighbors, and how the individual person with the diagnosis experiences that diagnosis is intimately related to their relationships with other people and vice versa. And I think we need to move beyond just putting individual people into research studies and think about families as well. Because if we don't have a better way of supporting families, then it makes life extremely difficult in terms of those families really supporting their children. So we've done some research looking at particularly parental mental health, and trying to understand whether the specific genetic diagnosis made usually in a child or in a teenager, whether that specific genetic diagnosis has some influence on parents' mental health as well. And moving on to do more work, which we're not going to talk about very much today, but more work on actually what it means to adjust to having a genetic diagnosis applied to a member of one's family. So those are our 3 research priorities: mapping the variation within genetic disorder groups and understanding that, trying to get under the surface to start to understand what is different about brain development and brain function in these conditions, and also relate that to the experience of parents and siblings and families as a whole. So those are our research questions. We are really interested in whether our research questions, which are motivated as clinicians and as research scientists as well, who are interested just to understand much more about genetics. We're really interested in knowing whether these questions do match your priorities and the priorities of your communities. We may have some time to talk about that a little bit now, but also we'd be very keen to receive your written thoughts and questions and feedback via our project website, which is here. That's the end of my short introductory presentation. Does anybody have any responses to my questions here on the screen, or any other questions for me? No problem if not, I entirely understand.
Maybe we will have more coming in after all the presentations.
Absolutely. And this is really about starting a conversation with people who are providing some basic information about what we do, but also starting a conversation that can carry on over time with individuals and with groups. So that's me done there. I'll stop sharing and hand over to my teammate, colleague and friend, Anna.
Hi, thank you, Kate. Thank you. Let me just share my screen as well. Okay, you should be able to see it now.
Yeah.
Here we go, perfect. Hi, everyone. I'm Anna. I'm a research scientist working with Kate. And so I am leading on some of the recruitment aspects of the study and also some of the questionnaires that we do that Paolo will talk to you about his experience with afterwards. So what have we done in Bingo since 2016 is that we've recruited 175 children and young people and their families, and they've taken part in our questionnaire study. We've visited 15 people with 15 different genetic disorders and identified families across 27 different disorders. We have gathered detailed information about the achievements and challenges facing every participant and family. And we are still recruiting now. And then if you, what we do as part of the study is have parents or carers complete questionnaires and interviews about medical history, adaptive ability, social skills, emotional behavioral difficulties, and activity levels in their child or adult with a genetic diagnosis. And then what we ask of the children and young people to do is complete activities that assess their motor skills and cognitive skills, including attention and memory. So what I would like to do, and I know that there are many experts in this room from doing your own research and from kind of experience with this directly, but I'd like to talk about kind of the genes in focus and what unites TRIO, DNM1 and SYT1 together. So the synapses you can see here at the very top, the red loop is that the process is called synaptic vesicle cycling, and it's to do with release of neurotransmitters, which helps brain cells communicate between each other. It is fundamental for cell-to-cell communication and underlies activity-dependent neurotransmission. So mutations in genes that affect this cycle don't mean that the brain cell itself is compromised or that it's not developed. It just means that the communication between brain cells may be impaired or slowed. At the moment, we have identified 33 genetic disorders in association with disruption to the SVC subprocesses. All of these 33 disorders share a common set of neurological symptoms, including cognitive impairment and intellectual disability occurring with neurodevelopmental difficulties or mental health difficulties. So what we know so far is that we have recruited 22 families with SYT1, 5 families with TRIO, and 2 families that have had a DNM1 diagnosis into the study. And what has been identified by previous research groups and by this research group is that the result of disruption to any part of the synaptic vesicle cycle are intellectual disability, movement and disorders, vision problems, and social emotional difficulties. So, however, there's a wide spectrum of ability, and there's different strengths and different challenges faced by each individual that we are trying to look into. So the next bit is that as you can see here, because we do have 22 families with SYT1 identified, this is our biggest group at the moment, and we can kind of look in depth into what the behavioral profile is of this group and try to understand whether we're capturing this adequately with our questionnaires. So what we have here is a graph to show the different adaptive ability skills in children with SYT1 versus kids that don't have an SYT1 diagnosis. So this is the non-synaptic group, but also have mutations in a gene that leads to intellectual disability. So as you can see here, the dark purple line is the SYT1, that communication and motor abilities are statistically significantly different and lower from the non-SYT1 controls. What we also see is that there's a large difference in functioning. So these bars, the black bars, just show the range of skill within each group, which is quite wide. So in the future, what we hope to do is to increase the sample size for all the other genes discussed and compare them across syndromes. The other thing that we can do is we can break down the bars individually and just look at, rather than the group average, to understand the range of individual variability. So this is the average of the same plot that I showed you before. So as you can see, the SYT1 group with the diamond in the middle is a little bit lower than the ID controls. But what does that actually mean for the individuals? So this is the SYT1 group broken down into the different genetic mutations. And as you can see, some of the individuals have their abilities and skills go up to 80, which is normal kind of level of functioning, to as low as 20. And there doesn't seem to be a very consistent pattern at this moment, and due to the number of people we have in each of these mutations, we ideally would like to increase the sample sizes to understand what the link is between the mutation and this profile. But we can see that some dimensions might be more useful to understand than others, such as communication and motor ability, as well as visual function, which may be kind of the key under the surface dimension to higher order abilities, like communication and cognitive development. There is also a wide range of ability in the control group, as you could see, but we want to also understand what can explain the individual variation. So what does all of that data mean? Is that there are features that are shared across both groups. Some things may be different, but there are similarities. But every person within each group is unique, and we are interested in understanding why. So as Kate mentioned before, unfortunately, there isn't a nice direct link between a genetic mutation and behavior. We have to understand what's going on in the brain and how those early mutations, how they affect how the brain develops, how these skills develop, and why there may be such large variability. So it may be frustrating at the moment not having the information in terms of what's specific and what's characteristic of the diagnosis, but with your help and with the help of the families, we are a little bit closer to that now, and we are able to, per each syndrome, we're able to produce publications, which is great, but it's trying to move that forward and try to create a united group that we can understand it a bit better. We do want to get away from kind of expectations of clear differences between groups and understand what's the shared patterns of symptoms and what are the individual concerns. So what we are also doing to help with the brain, understanding of the brain, is designing a study for neuroimaging in the home. There have been some pilots done previously, but what we're trying to do is design a study that will work for a whole range of the genetic disorders that we have, or kind of 33 or more of them, that also would be appropriate for individuals with severe levels of intellectual disability. At the moment, any studies with MRI and MEG aren't fully appropriate for patients with severe intellectual disability, which means that they have unfortunately been left out of some of the fundamental neuroscience research. So trying to get to understand that and how we can do it in a non-invasive way, in an easier way, in a mobile system that can be done in the home is our next move. And then hopefully we'll be able to understand a little bit more about brain behavior and how the genetic side of it impacts everything. Yeah. And so how does your involvement help? So we'd like to understand your main concerns for families, to try to understand what the focus of the research should be, and treatment innovation, to help understand the strengths and difficulties, and also kind of try to get feedback and try to improve awareness in the medical community, as well as connecting families to each other, which is a very important aspect of all of these studies and of RareConnect itself. So if you would like to get involved, and if you haven't already, because I know that some of you already have, which has been fantastic, is that you can go on our website, which introduces the study, that at the moment we've got online questionnaires and online parent-child assessments with future possibility for home assessments in the UK. And then my email is below the study email, so that you can send us any comments, but I think that will be provided for you after the talk as well. And I just wanted to say thank you to our participants, sponsors, and all the families listening today. Thank you. Yeah, and we've got the Q&A for the family discussion. Should we want to do that afterwards?
Thank you both. I guess we can invite Mr. Paolo Mancerini to join us and tell us a bit of his experience. And then we can, I see there are many questions, and I'm happy to either to read them to you afterward when we start with the Q&As, or I believe if people that posted those questions, if they wish to speak up and tell it vocally, it's not a problem. So does Mr. Paolo Macchiarini is here?
Hello,
okay.
Can you hear me?
Yeah.
Can you hear me?
Yeah.
Okay.
So
Thank you very much, Sandra.
Thank you for joining. I believe that we will all be interested in hearing your experience from the actually parents' perspective. And as you took part in the study, I just want to say one word more, that Mr. Paolo Mancerini is a moderator of the trial-related intellectual disability community on Rare Connect also. So I pass the floor to you, Paolo.
So, thank you again for the invitation. And to be honest with you, I didn't have time to prepare any presentation or anything because I was very busy this day. But I would like to give you my personal. I completed the first phase of the project, and yeah, I would like to share with you my experience. So basically, there are many modules. There are hundreds of questions you have to answer to. So at the beginning, it looked like overwhelming at first glance, but you know, then I start doing this. I start answering this question, and I realized that so the more questions you answer, it means that the more accurate is the investigation. And I was really glad that the question were going so deep you know, in understanding which is the behavior of the child, which is the interaction of the child with the society, with the family, and so on. And for me, this means that it's a really a serious study, and that's why I proposed this study to my community, to my friends, and so on. And also, there is another aspect I really appreciate, because answering the question, I realized, or better, I remember that something was wrong in my son, because basically we live with these children every day, and at the end of the day, we get used to their behavior, we get used to their habits, and so on. So, I mean, we love these children. We wouldn't change for. I wouldn't change my son with any other child, even if he's let's say a little bit problematic. But I love him, and really he's my angel, and I wouldn't change him for anyone. But sometimes we really risk to get used to this behavior, and it means that we don't insist in doing something in anticipate something, mitigate the situation, and but answering this question, really, I started putting down all the point where I have to work to improve the situation of my son. So this is also something I really appreciated by doing this study.
Like, come in. Thank you so much, Paolo. It's Kate here. It's wonderful to meet you, even online, and we're so grateful for your support and your comments and feedback, and your membership of the project. And this kind of research is something that we have to do together, and it's really helpful for us to know that the initial your initial feelings about taking part was initially it felt overwhelming, and it's also very helpful for us to understand that the experience of answering all those questions actually themselves was in the end, something positive for you and for your family. So we're really grateful for you sharing those experiences with us. Thank you.
It's a big pleasure for me, Dr. Kate, to meet you. And yeah, thank you.
Also, listening to you also drew so many of the really fundamentally important questions and I guess balances that we are hoping to strike or to help with, because developmental disorders are so complex, and they raise so many different questions at many levels about what we expect from children and children's development into adults, what our hopes are for the future, how we balance their strengths and their magic, unique impact that each child has on your lives as parents and on the Ryder family and on all of their friends, how we balance that love with wanting to help and support them so that they can achieve everything that's right for them in life. And those are complex questions that are different for each child, different for each family. And we're not afraid of that complexity, but I think it informs so much of what we do, that we have to be as kind and gentle as we can in our science, and not make assumptions about what everybody wants or needs after a genetic diagnosis. Okay. Does anybody have, sorry, I'm taking, Sandra, you're moderating. Over to you.
That's fine. You can take all those questions. So thank you, Paolo. And it was lovely to hear from you. And I already see it will be a fruitful discussion. So I'm going to read questions from the chat box, but please, those who posted them, you can cut me out at any moment if you want to speak out or add something to what you wrote. So Amanda asked, I'm very interested in what information will be found for life expectancy for those with a trio mutation, and what other health, sorry, I have a small screen here. And what other health risks will, apologize, I have some difficulties with Windows. And what other health risks will happen over time?
I can briefly answer, and then I'm sure Anna will answer some of the other questions too. But so as part of the Bingo project, as Paolo mentioned, there are a number of different questionnaires, one of which is a medical information questionnaire. So we've tried to collect the same information about each person in the project in terms of what happened when they were very first born, in the first few years of life, in early childhood, teenage years, and as they grow up. And when we produce our research papers, we tend to summarize that information so that there is a better resource for families and for doctors and other professionals when they're seeking that kind of information about medical problems for each genetic diagnosis. But on our own, this project can only do so much. So I think it's always really important that the information that we collect and summarize and report is also looked at alongside other research by other groups, and only by kind of gathering information from multiple different projects over time will we really build up that picture. I think it's particularly important that we involve as many teenagers who are diagnosed or adults that are diagnosed, and in a sort of a different setting, encouraging clinical services to offer genetic testing to adults with intellectual disabilities who have no diagnosis, because of course the older people are when they're diagnosed, the more useful information we can gather for younger children now. So it will just take time for that information to emerge.
Thank you. If Amanda has something to add, please unmute your microphone. Okay, so I guess we can continue. One question from Mark Dooms. Why are there very few orphan drugs authorized to treat rare psychiatric disorders? Is the blood-brain barrier a problem?
Such a great question, Mark, and I'm sure you're aware there are so many different reasons. I mean, until the last, say, the number of genetic diagnoses that have been discovered has very, very rapidly increased over the last 10 years. So there is such an enormous sort of tidal wave of new disorders that the scientific community and the pharmaceutical community are just way behind. But there are increasing numbers of initiatives to tackle this challenge, really, and to come up with approaches that are going to be relevant to the rare disorders community. So one thing that I'm involved in is trying to think about pathways towards treatment which will be relevant to groups of rare disorders. So as Anna just mentioned, the reason that we are looking as a group at TRIO and DNM1 and SYT1 is because those genes perform similar roles in brain cells to do with neurotransmitter release. And we're thinking that if we can understand those disorders as a group because they're linked by a similar mechanism, then that increases the number of people for whom a potential treatment would be relevant. And it also would increase the possibility of really understanding those mechanistic links between the genes and the actual symptoms and day-to-day problems that people need to be treated. So that's the approach that we're kind of starting to take. But again, it's about having patience and time. Other basic reasons that the pharmaceutical industry, by and large, hasn't been interested in neurodevelopmental disorders, or even mental health in neurodevelopmental disorders. It's just not been high priority, but that's changing. And then I'll come back to this really big complex question, which is thinking on an individual level and at a syndrome level, what are the real priorities for treatment? What would make the biggest difference to the quality of life of patients with these diagnoses and these families? Sometimes, for example, I know that research scientists might be very focused on epilepsy treatment, for example, which is very important, or might think about things that could improve cognition by a little bit. Well, and by and large, seizures mostly, not always, but mostly actually they're quite treatable by existing drugs. Some patients, it's a massive problem, but usually you can get those seizures under control usually. Whether there's a drug that in an animal model will make a little bit of difference with a little bit of IQ points, well, the question is, would that really translate to the kind of impact that your children and you really need? And that's why so I basically think that we have to think toward treatment on these multiple different strands. And fundamentally, we first just need to understand these disorders, understand what the core of the problems really are, what are the things that really matter in day-to-day life, and those will be different for each person, and what are the things that we could safely modify about brain function, where we would really make day-to-day life easier and not make problems worse. So it's not that we don't want to get that way, that's our direction of travel, but it takes many different lines of research in parallel, I think. What do you think, Mark?
I agree, yes, but we can repurpose some medications to treat mental disorders, like psychiatric disorders, for example. Wouldn't that be a good idea?
Yes, and thank you for mentioning it. So I guess the one example that I can give you so far of how our research in this area of synaptic vesicle cycling disorders has moved in that direction was that when I first, and my colleagues first described SYT1 related disorder, the biggest problem that we saw was affecting the first patients that we diagnosed was involuntary movements and being awake all night, very unsettled, on the go the whole time. And that it wasn't just the sleep problems, which are huge, but these uncontrolled movements. We were studying the mechanism by which SYT1 was leading to those movement disorders and thought, what drugs do we already know exist that aren't usually used for children, but are actually used in elderly care for elderly patients with Parkinson's disease. And we thought, based upon the mechanism, how we thought SYT1 was causing those symptoms, the neurologist looking after that particular child actually repurposed a drug that is a licensed drug they wouldn't normally use. And it was extremely effective, and the patient is still well on that drug now. So that's my one kind of single case study of how discovering cause, understanding the mechanism led to a new treatment and better symptom control. But it didn't take a drug trial to do it. And so we should be doing that more.
Thank you, Mark. I'll continue with our questions. Paulo asks, dear Dr. Baker, is it possible to create a model to predict neurodevelopmental outcomes, cognitive delay, psychomotor issues, neurosensory problems, to mitigate potential risks and choose the right therapies?
And as I put in the chat, Paolo, I'm so grateful for you asking that question, because I've literally just been writing a funding application with a computer scientist and a neurobiologist at Newcastle University to attempt to build a model that would do that. And it's such a crazy idea. So I'm so encouraged that you think it could potentially be a useful approach. So we probably won't get the funding for that, but I'm enormously inspired and grateful for your question, that we will try. And I promise that I will try to build up. Also, I'm not a computer scientist or somebody who understands the language of models. So it's a massive challenge for me. But I think my role and our role and Anna's role in this project is to try and translate what we've learned from, say, the questionnaire study, something totally different, and from listening to you as families, try and translate the reality of your children and the day-to-day reality of human beings in real lives, and think, first of all, well, what are we trying to predict? What do we want the outcome of this model? And try and communicate with the computer scientists and the basic biologists who can do experimental things with cells, nothing to do with humans, and translate from the experience of the living with these disorders to a useful model. So we'll try.
Yeah, and absolutely.
And you can help us, please.
By gathering data, we're able to make the models more informed, which is, so these things are very important rather than, and as same as treatments, we need to understand sometimes what the concerns are, what the variability is, to try to understand what we can do.
Absolutely, and just following on from that with Anna and Anna's communication with families via email or over Zoom or phone, that's such rich and important information that can make sure that when the scientists go up to the lab or to the computer simulation system, that they're really going to be trying to address the questions in the way that will be valuable for you. What we want to do there, to go into a little more detail, is based on our experience with SYT1, where we know a number of different variants and this quite characteristic set of problems with movement and vision and communication, but that varies a great deal within that syndrome. And we would like to know how much information about the specific variant, but how much information about the effect that that variant has on cell function. If we put those bits of information together into a model, would it both predict the problems that the individual person has, but also, as you said, would it point toward what sort of brain mechanism might be the right kind of target to treat to improve those symptoms? And I think it's going to take a very, very long time. But that's the way we're starting to think. We'll try.
Okay, we received one more question from Amanda. Am I to understand that your team is focused on collecting data to drive future medical research?
Well, yes, we consider this to be medical research. I'm a medical doctor, and I know that this information in itself already will translate to better medical care for patients, because here and now in clinical genetics departments around the world, diagnoses of trio or of DNM1 are being made, but the evidence base that doctors can use to give information to families is just not of a good enough quality to really inform prognosis and medical care. So the information itself, the information from questionnaires, once we get enough patients participating, and Anna can analyze it effectively and compare it to other genetic diagnoses, because it's so important to put our observations about each specific genetic disorder in the context of what we expect for developmental disorders as a whole. So we can really get to what is specific about each genetic diagnosis and what is not specific, very important, but not specific to that diagnosis. So I see this as still very valuable medical research. In terms of where it then branches out from the starting point, it's in the number of different directions. So understanding brain development, thinking about underlying brain mechanisms, which would inform different treatment strategies in future. That's the ways that it branches out from this point in time.
Thank you. Is there any more questions to ask our speakers?
Can I ask one question?
Yeah, of course.
Okay, so there is a typical question from my community, from people of the community. So they're always asking, is bingo an international project? I mean, there are several steps of the research that involve also, let's say, a face-to-face meeting with your team and so on. So some people are wondering how is possible to go ahead to move forward in the research if we are not, if it's not possible for us to travel to Cambridge and so on. Thank you.
Anna, would you like to answer? Thank you, Paula, for the question. Anna?
Thank you, Paola. Yes, I can. I think we've discussed this a few times as well, but the project is international. We have, due to the recent COVID pandemic, we actually have been able to make our study a bit more online friendly and to increase those numbers by trying to reach out to different communities. So we have patients from many different countries now. And the map that I showed had all of the ones that we've identified people at and that have provided questionnaire data. The questionnaire data itself is very valuable for us. It puts it into context in terms of what we should expect, and it gives us a lot more. It gives us our studies and our kind of findings a lot more power and credibility because we have these large sample sizes. We are trying to move forward with online assessments, which will look at movement and social interaction that we will be able to deliver over a online video platform. We haven't decided on which one, but there are some options that make sure that the privacy of families is respected, that are also used by the NHS. So this is our National Health Service. So hopefully we will be able to get that up and running for international families. In terms of the UK studies that we have and the visits, they are very small sample kind of studies because it's not that many people in the UK actually have these diagnoses, but actually we're able to get a lot of data from those families within kind of the neuroimaging field. And then hopefully, because we would be doing that and because we are increasing awareness and we are increasing the number of people that are involved, other countries and other research groups see it as a priority to do so. We are also we're collaborating with people in Australia who are interested in the synaptic vesicle cycle, who are primarily work within genetics and things like that. And then we have identified patients. So there are those links that are being built up now, which is fantastic, but we are actively working on that.
rare disorders research has to be global and flexible and creative and collaborative, but also realistic. So we're doing what we can globally. We really need to make sure that it's accessible, perhaps more accessible to non-English speakers. It's a definite weakness in what we do at the moment. So we need to just really make sure that we increase access as much as possible. And never say never about us being able to travel outside of the UK. In the old days, crossing over into Europe wasn't as difficult as it is now. And maybe there will be time again in the future where we can be more mobile and see if we can cross borders. No promises, but let's hope for that in the future. Okay, I think it's nearly 4 o'clock.
Yes, and I'll ask one more time if there are any questions.
Last question, I promise you, this is the last question from my side. I promise you. So, one question. For instance, my son is affected by trio gene mutation, and now let's say we found which are the labs or the research group that are studying deeply this gene. For instance, there are labs correlated to Yale, another labs correlated, another lab correlated with
He might have some issues with internet connection.
I think probably time to conclude, and we will make sure that we contact Paolo. We'll contact you for your question. Anna will make sure that we do. And never promise that it's the last question.
Okay, so in case nobody else wants to ask any more questions, I believe we can conclude this meeting. Prior to that, I would also ask Dr. Kate, Dr. Anna, if there is anything to summarize or emphasize that after this fruitful discussion, maybe
just that we're so
came
grateful
out.
for, we're so grateful for you all taking the time to join the webinar today and for all of your thoughts and contributions so far. We know how busy and how challenging life is day to day. We really enjoy the work that we do, and if you would like to stay in touch, we'll always be delighted to hear from you.
Yeah, absolutely. Have a look through the website link and just email us if you have any questions. We'd love to talk.
Okay, bye-bye, everybody.
Okay, so just one more sentence from our side. We will be sending you the webinar recording shortly. And of course, if you still have questions after releasing the recording, or if you think of something important that is missed to address during the Q&A part, please either reach out to our speakers, or you can also contact us via info@rareconnect.org, and we will make sure to pass the questions to speakers and get back to you once we get answers from our speakers. So thank you once again for joining. I hope that this webinar was helpful to all parties, and see you in some other organizing other meetings and something similar to our patients.
Thank you. Sorry, that was my dog.
Thank you all.
Thank you, Sandra.
Thank you.
And Sandra and Melanie, if you have feedback for Anna or